Good morning, everyone. Thank you for being here during the Neurospirits of this week. And I would like to using Parul Verma from the University of California, San Francisco, and more specifically the Raji Lab, which today will give a talk about biophysical spectral graph theory based on for model for brain oscillations. So thank you, Parul. So if you want to start, the floor is yours.
Great. Thank you so much for having me here. Very excited to present this work. Today, I'm going to talk about a biophysical spectral graph theory based model that tries to capture the spectral patterns of brain oscillations that we can capture empirically using MEG or EEG. All right, I'll start with giving an overview of this model. The way it works is there are two levels. There is a local mesoscopic level where we model the local excitatory and inhibitory neuronal subpopulations, and all of them get some input white Gaussian noise. And that goes as an input to macroscopic long range excitatory pyramidal neurons that are connected to each other via the structural connectome. And when we simulate this model, we get a plot of power spectral density, which tells you the power of each frequency, and which for every brain region, where the brain regions are parcellated according to whatever parcellation scheme we want to choose. So every line here corresponds to frequency response of a particular brain region. So, and this is what we get by simulating the model. And the nice part about this model is that you get this, the solution in a closed form in the frequency domain. So you don't have to do long time simulations to obtain this plot. It's very quick to obtain this frequency response from this model. So it's a hierarchical model with two levels. The first is the local level, and it has a small set of parameters. You have some neural gains that control the gain of the signals. And then you have some time constants, tau e, tau I, and tau g, that characterizes the time constant or the rate of change of the excitatory and inhibitory populations. And tau g corresponds to the excitatory time constant of the entire macroscopic graph. And the local population gets white noise, and the macroscopic long range connections are constrained by the structural connectivity matrix, which you obtain from diffusion MRI. Now, there's also a coupling constant in this model corresponding to alpha, which controls the extent to which different regions are coupled to each other. And then you have another parameter for speed, which regulates the speed at which different signals are transmitting from one brain region to another. So the nice part about this model is that you have a very small set of model parameters, and you get the frequency spectra in closed form, which you can directly compare to what you get from magnetoencephalography or MEG. And you only need a small set of global parameters to obtain a frequency spectra that accurately captures empirical MEG. So there are two things we've tried doing. We've tried to simulate this entire model, keeping all these model parameters as global, which means they are the same for every brain region. So we have a very small set of model parameters to infer, and we obtain a frequency spectra for every brain region. And the other thing we have also tried is to just look at the local model and try to fit the local part to every brain region separately and estimate these model parameters for every brain region separately. And both of them can be done easily, and parameter inference is very tractable in both the cases. And I will show you applications of both of them in the subsequent slides. All right. So with this background of how the model works, I'm going to talk about three different aspects of this model. First, we'll go into model fitting, where we'll talk about the paper on spectral graph theory revisited and improved. And then I'll show you some of the applications of this model to see how we can apply in clinical and biological settings. And I'll also show you some of the theoretical advances we've done with some theoretical explorations with this model. All right, let's go into the model fitting part. So the main idea was that we obtain a frequency spectra and we want to make sure that it matches the empirical spectra we obtain from MEG. While doing so, we estimate what the model parameters are. So we did that. That was the first thing we did. We tried to see how the spectral patterns fit to empirical MEG. And for this, we obtained MEG recording from 36 resting healthy subjects. And that's what you see in black here. The black line corresponds to the MEG recording of 36 resting healthy subjects, averaged over all subjects and averaged over all brain regions. And in blue, you see the fit of SGM to the MEG spectra. So you can see the fit is pretty good. It captures the peak and the fall off and accurately. And the Pearson's correlation has been greater than 0.9 for most of the subjects. So we showed that this model, you get a closed form solution in Fourier domain and very quick with very less simulation time, and you can accurately capture the frequency spectra which you obtain empirically. So that's about spectral fits. What about spatial fits? So for spatial fits, the first thing we tried doing was that we sorted the eigenmodes we obtained from this model in a way that whichever ones lead to maximum spatial correlations, they go first. So this plot is a plot of, on the x-axis you have the sorted eigenmode index, and on the y-axis you have the spatial correlation of the alpha frequency band. So we are only focusing on capturing the spatial pattern of the alpha frequency band, and we try to see which eigenmodes are most important to which that lead to maximum spatial correlation. And to give you a visual idea of how that looks like. So this is for a particular subject. This is how the alpha frequency band spatial distribution looks like empirically. And this is how it looks like when we include the top eigenmodes from SGM. And on the right, this is for a different subject. This is how it looks like from MEG, and this is how it looks like with our model. Now, the interesting thing was that the eigenmodes that led to maximum spatial correlations were not the same for every subject. In fact, they were widely distributed, as you can see in this bar plot. We did a similar exercise for beta frequency band, and we sorted the eigenmodes that we obtained from SGM, and we plotted and we looked at which are the top eigenmodes that lead to maximum beta band spatial correlation. And to give you a visual idea, this is how the MEG beta band spatial pattern looks like, and this is the one from SGM for one particular subject, and this is the same comparison for a different subject. Again, we saw that the top eigenmodes that lead to the maximum spatial correlation are widely distributed. It's not the same eigenmode which leads to maximum spatial correlation in all the subjects. So then next, this was even without trying to fit for spatial patterns. We were our cost function was still just a spectral correlation between MEG and SGM, and this was just an outcome of the model without trying to fit for the spatial patterns. So our next question was, can we try and maximize for spatial fits as well as spectral fits together and try and estimate model parameters? So that's what we did next, and we added a spatial correlation as a cost function in our model, and this time we are fitting to just the alpha band spatial patterns in addition to the spectral patterns. Interestingly, we saw that we can fit to both. This is a case where our cost function was spectral correlation plus spatial correlation, and we saw that the spectral correlation as well as the spatial correlations were high. While if we just keep one of them as the cost function, If we just keep, for example, spatial correlation as cost function, the spectral correlation goes down. And if we just keep spectral correlation as cost function, the spatial correlation goes down. So we came up with a way of jointly maximizing both spectral as well as spatial correlation using our method. And to give you visually how it looks like, the empirical spectra matches the MEG spectra. The empirical MEG spectra matches the SGM spectra. And this is the averaged spatial distribution of alpha frequency band in MEG and the average spatial distribution of alpha frequency band in SGM after maximizing for spatial correlations. So using this, now we have a model of SGM which can try and maximize spectral as well as spatial patterns simultaneously. All right, so that's good. Our model can do some can be used for fitting, and we can infer some parameters. Now the next question is, can we apply to any clinical and biological settings and gain some biophysical insights from a model? So for this, today the work that I'm going to show you is going to be only on the local part, where we focus on just the local excitatory and inhibitory signals in different brain regions. And for every brain region's spectra separately, we estimate what these model parameters are. And I'm going to apply this model parameter fitting to two cases. The first is going to be on Alzheimer's disease. And so to summarize what we're doing, we obtain the MEG frequency spectra for every brain region, and for every brain region we apply the local part of SGM, and we try to fit to the observed spectra. When we do that, interestingly we see that the model parameters are differentially distributed in controls versus patients with Alzheimer's disease. And specifically the time constants, the excitatory and inhibitory time constants, the effect size was pretty large in both both excitatory and inhibitory time constants. So that was interesting. And in addition, we saw that if we look at how the model parameters are correlated with tau and amyloid beta, which are molecular markers of Alzheimer's disease pathology, we see that the excitatory time constant and the inhibitory time constant, they are correlated, significantly correlated to tau and amyloid beta. So from doing this exercise, by doing this kind of parameter fitting, just by using a small set of model parameters, we can get some biophysical insights into any disease that we are looking at. And by seeing that there is a difference in the excitatory and inhibitory parameters here, we can say that there is some sort of excitatory inhibitory imbalance in Alzheimer's disease that is indicated from this differential distribution of model parameters. So that's the application to Alzheimer's disease. We also applied the same model to look at different stages of sleep. So here W stands for wakefulness, and N1 and N2 are two different stages of sleep. We again looked at we again tried to fit our model to MEG recordings of subjects going from wakefulness to different stages of sleep. And again, we saw a differential distribution of model parameters indicating a change in the excitatory inhibitory balance when we go from wakefulness to different stages of sleep. And this is just to show you the model fit. This is how the actual empirical MEG spectra look like for different stages of sleep, and this is the one we got from our model. So you can see there's a close match here. All right, so all of this was about applications, just to give you a sense of how we apply our model to gain some biophysical insights. Now, we also wanted to understand this function of this model better, and in particular look at how when this model is stable and when it's not, and then extend it to look at some dynamics. Because so far we have looked at the static frequency spectra. We wanted to see, can we extend it to look at how the frequency spectra varies with time, and then how model parameters vary with time to get some insights into dynamics of short scale dynamics or some sort of micro state switching that can give us that can also give us some biophysical insights. So to start with, we looked at the stability of this model, and we looked at stability in two stages. We looked at the stability of the local model first, and then the macroscopic model separately. So I'm going to show you how this local model looks like first. And to understand how stability of the model is, we first try to simulate the model. So if we simulate the local model just without noise, we see that there are three regimes possible. The first regime is stable, where the amplitude of oscillations die down. Then you have a regime where it's borderline stable and you have limit cycles. And then it goes unstable and the amplitude of oscillations explode with time. And we get these different regimes by varying the model parameters. So for some model parameters, the system is stable, and for some it's unstable. With noise, when we add noise, it becomes a little unclear, but you can clearly see that when it's unstable, you can look at the simulations in short time and see that it's unstable. So given that there are different regimes possible, the next question we asked was, can we obtain stability boundaries? So that's what we did. We did a stability analysis of our model equations to obtain borders of stability. And this is a plot of GII versus GEI, the two gain terms. And we see that depending on what the time constants are, we have a region where the system is stable and we have a region where it's unstable. And interestingly, we looked at how the frequency spectra looks like just in the stable regime. And we see that we get a variety of frequency responses, in particular if you change the time constants. So for example, if you increase tau e, the peak frequency first is around alpha band, and then it goes down and the peak frequency goes down to a much lower frequency. And similarly for tau I, we see a same pattern. Well, for GII, the second peak, if you increase the GII, the second peak goes from a smaller frequency to a larger frequency. So this plot is just to show you a variety of frequency responses that we get within the stable regime if we sweep across the parameters. Now we did a similar exercise of stability, but this time looking at just the macroscopic model alone. Again, to get a sense of the model behavior and to get a more theoretical insights into how this model works. And this is how the stability of the macroscopic model looks like. In plot A, I have a plot of tau g, which is one of the time constants, versus alpha, which is the coupling constant. And we see that we have different regions. We have this region which is stable, and this where it's unstable, and here where it's unstable. So if you look at a plot from the stable regime, which is indicated by 1, that's shown here, by stability, what's happening is the oscillations are dying down with time. If you look at the plot in 2, which belongs to the unstable regime, the oscillations are increasing with time. And for both 3 and 4, the mean of oscillations are increasing with time, and that's why it's going unstable. So this summarizes the stability of the macroscopic model in total. Now, another interesting thing we saw was we again looked at how the frequency response is of this macroscopic model alone, without getting the local model as an input, just to see alone what it's capable of producing. And what we saw was that this is a plot of tau g versus alpha again, and the color indicates the frequency at which the peak of the frequency response is occurring. So for example, if you look at green here, the green color indicates that the frequency response peak happened around 2 Hertz. And these colors indicate that the frequency response happened around, the peak of the frequency response happened around this signals. So happen around this frequency. Now, the interesting thing about this is that these frequencies belong to the alpha frequency band. And so even without any local model input, we can obtain a peak in the alpha frequency band just by the macroscopic long range connections. So this shows the richness of the model and how it can obtain uh, 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 empirically realistic peak frequencies just by even without any local model going as input to the macroscopic model. So with this, we did the stability to, go into to get a sense of how this model works, but also to see can we track how the stability of the model is over time, and if we can get a sense of how stability changes with time, which can also be applied to different diseases and brain states. And that's what we have started exploring now. With this entire model, we have started exploring how the model parameters can change with time, and in turn, can we track stability with time. And this is how we do it. What we do for this is we obtain time frequency decomposition of the MEG recordings for different subjects. So if you the x-axis here is time in seconds, y-axis is frequency, and I'm just giving you a plot of 3 different subjects out of the 36 subjects just to give a sense of how the time frequency decomposition looks like for different subjects. And what we do is from here at different time points, we extract the frequency response and we try to fit our model to the frequency response. And then we get a temporal distribution of model parameters. So for example, we extracted a frequency response at around 10 seconds, and then we tried to estimate how the model parameter looks like at that 10 seconds for different subjects. So here the bottom plot tracks how the model parameter alpha varies with time. Here x-axis is time for different subjects, where y-axis corresponds to every row corresponds to a particular subject, and the color indicates the value of alpha. So what you see here is how alpha is changing with time for different subjects. And we did the same for the neural gain parameters GEI and GII, and we let the other parameters be constant over time. So what we see interestingly here is this alpha, which is a coupling constant. You see a stark switch from a low value to a high value, where a low value is indicated by a darker color and a high value is indicated by a lighter color. And so we see a stark switch between a low coupling to a high coupling value. So we found this interesting, and right now we are trying to see if we can correlate alpha with the extent of integration and segregation, because if it does, then we have a very parsimonious way of capturing integration and segregation and how it changes with time using our approach. And with this, we were also able to track how the stability of the model changes with time to get a sense of how stability of brain oscillations might be changing with time. All right, so this is all I wanted to present today. There were 3 aspects I wanted to present about the model. The first was on model fitting. We showed that this model can be fit to empirical MEG, and we get high spectral as well as spatial correlations of alpha frequency band, shown in a summary figure here. And then we applied the local part of this model to Alzheimer's disease and stages of sleep to show how the model parameters are differentially distributed in control versus Alzheimer's disease and in different stages of sleep. And it gave us an indication of how there might be an excitatory inhibitory imbalance or a change in excitatory inhibitory balance at different stages of sleep and in Alzheimer's disease. We also did a stability analysis to get a more sense of how this model works and what are the parameters when it's stable and when it's not. And then we extended this work to look at how we can track model parameters varying at different time points in a feasible parameter inference way, in a way where parameter inference is feasible, and we track how the dynamics look like. And now what we want to do is we want to investigate integration, segregation, how it varies with time, and we also want to see can we obtain this kind of dynamics of model parameters for healthy and different diseases, and see how these dynamics are different in different diseases as well as brain states. So there are a lot of people involved in this work, but I wanted to highlight my PI Ashish Raj, my co-mentor Srikanth Nagarajan, and Kamalini, who spearheaded the Alzheimer's disease work, and Jolene spearheaded the sleep work. So that's all I have for today. Thank you for paying attention, and I'm happy to answer any questions you have.
Yeah, thank you. I hope the remote clap arrives. Yeah, I have. Okay, before there are any questions, but I have maybe two as a starter. So just one thing, when you say change of segregation integration, you mean within the same session, like a dynamical?
Yeah.
Okay, this was the simple question. Then maybe the one can you give more details when you were saying that there was the correlation with PET biomarker and excitatory time constraint?
Yeah.
So okay, so there are two biomarkers, one for tau and one for AB, localized in. So this is the brain spread.
Yeah. Yeah.
So how were you doing this this correlation? Like the local, the region? Is there an atlas? How is this? Is an average of voxel wise? How is this?
So we parcellated we didn't look at voxels, but we parcellated the brain regions using Desikan-Kiliani atlas. And then for every brain parcel, we estimated the model parameters, and the tau and A beta was also parcellated according to the Desikan-Kiliani atlas. So for every brain parcel, we tried to find correlation of time constant versus the tau and A beta value in that brain parcel.
And this was, so one thing we noticed is that the tau is they're generally increasing, while the A beta, they increase and then they go down. I'm talking PET in general. So in this data set, you had both cases of tau increasing and then decreasing, or what? Because I don't think this is the ADNI, because you have MAG, so I think this is data you acquired.
Yeah, and this is static data. We are not looking at change of tau or amyloid beta with time.
No, but the problem is that depending where you're catching the guy.
Okay.
So when you're doing the scan of the person, is either is on PET, is increased. There are these stages of Alzheimer.
Right.
So I was wondering if you have enough heterogeneity. You probably, if you did one time, you probably don't know.
I don't know about the heterogeneity part. I think my the first author, she'll know more about that part.
Okay.
Oh, yeah.
No, because the A beta is we're studying is very tricky marker. So I was also wondering if there are different moments of Alzheimer and different correlation you find in the MAG or.
Yeah, I don't know about that. Yeah.
But if it's cross-sectional, you have one time point, you probably have no idea to know this.
Yeah. From what I know, it's a one-time point.
Okay. It would be nice if you check a different longitudinally if this thing is still.
Yeah.
It
Yeah,
seems
that's an interesting point.
It seems you have quite straight lines. Like it seems whatever is correlating.
Yeah, I'll bring this up and see. That'll be an interesting extension to look at how correlation changes with time.
Yeah, especially because we notice A beta is it's going up and it's going down. And the fact that it's going down doesn't necessarily mean the patient is better. It can also be a more advanced stage.
Mm-hmm.
Yeah, that was at least for me. Do you have other questions?
Well, I will jump in then. Can you speculate? Actually, I had a similar question like as this one. Can you speculate why do you have this strong correlation in this
Yeah,
case between amyloid beta and the excitatory time constant?
yeah, I think one speculation can be that uh, there is some relation between excitatory activity of the neurons and tau, and an independent relation between inhibitory activity of the neurons and A beta. So there's probably some sort of going on where the excitatory we don't know which impacts what, but there's probably some sort of maybe there's an alteration of excitatory activity impacting tau or vice versa, and independently uh, inhibitory activity impacting A beta or vice versa. But because these are correlations, we can't tell which is which. But that's what I would speculate. And that this plot also tells us that the inhibitory activity, there's no relation between inhibitory activity and tau. And so specifically, there's something with excitatory activity and tau, and vice versa with amyloid beta.
The correlation is for beta band activity. Sure.
Sorry?
This correlation of excitatory activity with tau and inhibitory activity with A beta is for beta band activity.
She's
What
asking if it's
means
specific for beta bands.
Yeah,
Beta.
it's specific for beta bands.
This part?
Yeah. Yeah.
This wasn't for beta band. No, like beta band doesn't.
Other bands.
So we looked at. So the constants that we obtained was looking at the entire spectra from 1 hertz to 40 hertz, 35 hertz. So it's not that we were only trying to fit to a particular frequency band.
Okay.
And you were saying this is EG and MEG, or this is MEG and you think can be
MEG.
applied to EG?
Yeah, this is
In the
MEG.
beginning you say this is MEG and EG.
We can apply to MEG and EEG both.
We can apply, okay.
Yeah, but so far we have only applied it to MEG in our work.
And how do you see like the vision for the future? How can we, not necessarily for Alzheimer, or how can we use this like in a clinical setting? You put a person and we do this analysis, and if the oscillation is certain thing, we suspect something. What is your vision? What can we do with this in the future?
We think that the work that we do is more applicable to generate research hypotheses than directly apply to clinical settings. For example, by looking this, we generate we see that there is probably some relation between tau and tau e and tau. So, can we in an experimental setting test for these relations? Or experimentally, can we test for some excitatory inhibitory imbalance?
And then from there, what does it mean? What can we do? Okay.
Yeah. From there, so we generate research hypothesis to understand the mechanism better. That's where we see our model can be helpful.
But do you think that this relation, because it's a very strong relation between the excitatory constant and the amyloid beta, maybe this excitatory constant can become a new biomarker for Alzheimer? I don't know if this makes sense. I mean,
Okay,
if
you have this picture showing, and we are only thinking about this.
No, I have other questions.
Maybe
I have other questions.
Yeah, actually, that's a great question. And we have started, so we applied, so this is just a local model, right? We, while we have this entire model with us as well, we recently did a fitting of the entire model to Alzheimer's disease versus healthy controls to see if we can get a parsimonious biomarker. And this is the work we are, this is something we are working on currently. And we saw that there's one particular model parameter which is different in Alzheimer's disease versus healthy controls, and it's also can predict extent of cognitive decline in Alzheimer's disease. So that work we think is something that can be seen as looking for a biomarker of disease. And that was more about biomarker because you get one particular parameter for an entire subject, while here you get a regional distribution of what the model parameters are for a particular subject. So I didn't show that work yet because it's not yet complete, but we're working on that right now.
Okay. Now the big picture is complete. More clear.
I don't know if anyone has questions, but I was just now curious. You build this model between regions, excitatory and inhibitory connections. I'm assuming that you between every region there are these two types of connections. But you talked about the stability of the model, and you only analyzed, I think, if I understood correctly, the stable region, right? So I was wondering if it makes sense to look at what happens in the unstable model in terms of biology. Does it tell something meaningful about, or it just like that the model is unstable here, you can tune the model here, and that's it?
That's a great question, and that's something we want to explore but haven't explored yet. So it's something we want to get to.
I have the impression that you may find something, at least in the close to the frontier. You also showed a picture where you some sort of had oscillations that were increasing. Right, exactly this one, right? But when you are super far away, it's just like a complete mess.
Yeah. So yeah, we want to do that, and we specifically want to track in a dynamic sense how the model stability is changing with time. And we're thinking of some applications here, like can we look at how stability is changing before a seizure onset? Maybe the model is becoming from stable to unstable. So those are some applications we're thinking of getting to soon.
Okay, yeah, sounds interesting. And the last one, I promise I will not bother you anymore.
No, go ahead, keep asking
What
questions.
about the phase space of the model? Like, did you look at it, or again, it doesn't make sense?
Phase space.
Yeah, I'm assuming there's a phase space. Maybe I did not understand that correctly. Like between the or no, I did get that wrong then. I'm assuming it's a system of differential coupled equations.
Yes, it's a system of
So
differential coupled equations.
If you look at the phase space of the model, does that make sense in some way? For example, the changes in the derivative against the variable. You know what I mean?
Yeah. We haven't looked at that so far in particular because we have so much focused on fitting to empirical MEG frequency spectra. So far we haven't.
Okay.
But one of our extensions right now is can we try to fit our model to, for example, amplitude envelope correlations, which is not the same as phase space, but some additional metric you can get just by looking at MEG.
Yeah, thanks for the great talk, by the way. Thank
Thanks,
you.
thanks for having me. Thanks for asking great questions.
So, anyone else? Oh, it seems we are all set. Okay,
All right.
was nice also because as I was telling, we are kind of following the work of Raji when they started with the simulation of spreading. Nice, interesting. And now you're moving into more biomarker from MEG and EEG. Yeah, okay. Then if there are more questions, we just thank you and yeah, I don't know if you have seen what we are doing. We are always open to collaboration. So if you or Raj had idea or visiting or any other things, we are open-minded to almost everything.
That's awesome. I'll definitely bring it with Ashish, Rajan. We'll see how we can take this forward.
Yeah, we just started the group, so we are not. I mean, his group has been around for a while. This has been just created, so we are just still in startup phase. Okay, it was nice talking to you, and yeah, maybe we keep in touch.
Yeah, for sure.
Okay, then thank you.
Thank you for having me.
Bye-bye.
Nice meeting everyone. Bye.